Hello, my name is Olivia Main, and I'm a student in Medical Physiology 1010. And today I will be discussing the short and long reflexes of the enteric nervous system. So the enteric nervous system maintains control over the gastrointestinal functions of the body. It controls motility, secretion, and growth of the digestive tract. It acts independently of the central nervous system and contains short and long reflexes. It has been termed the little brain in the wall of the gut because of how independent it can act of the central nervous system. So according to Silverthorne, the enteric nervous system was first recognized more than a century ago when a group of scientists noted that the isolated sections of intestine that were removed from the body created a reflex wave of contractions when pressure in the lumen was increased. So they observed the ability of the enteric nervous system to carry out a reflex independently of the central nervous system. And there are several similarities to the central nervous system within the enteric nervous system, including anatomical and functional features. So first we have the intrinsic neurons, and the intrinsic neurons lie completely within the wall of the gut, just like the interneurons are completely contained in the central nervous system. And the autonomic neurons of the ENS bring signals from the central nervous system, and those are called extrinsic neurons. And I will go into detail of the two types of intrinsic neurons on the next slide. And next we have the neurotransmitters and neuromodulators. So the enteric nervous system neurons release more than 30 different types of neurotransmitters and neuromodulators. They are most identical to molecules found in the brain. Among the best known GI neurotransmitters and neuromodulators are serotonin, vasoactive intestinal peptide, and nitric oxide. And these, the function of these neurotransmitters and neuromodulators are communication and integration of reflexes. Next, we have the glial support cells. So these glial support cells are more similar to the astroglia of the brain than the Schwann cells in the peripheral nervous system. And then the diffusion barrier is also a lot like the blood-brain barrier. The capillaries surround the ganglia in the enteric nervous system, and they're not very permeable at all, and they create a diffusion barrier. Like I said, much like the blood-brain barrier of the cerebral blood vessels. Next, we have the integrating center, and the neuron network of the ENS has its own integrating center, much like the brain and spinal cord. The reflexes that originate in the GI tract can be acted on without the neural signals leaving the central nervous system. So short reflexes of the enteric nervous system are the little brain aspect of this system, and they originate within the enteric nervous system and are integrated without any outside input. So this allows local reflexes to begin, integrate, and end completely within the GI tract. It receives a stimulus via sensory receptors in the lumen of the gut within the epithelial layer. The responses are carried out via enteric neurons and interneurons. So we have the submucosal plexus, which contains sensory neurons that receive signals from the lumen of the gut. The submucosal plexus controls secretion by GI epithelial cells, and this is how the ENS network integrates sensory information and initiates responses. The myenteric plexus neurons influence motility in the muscularis externa, and its main responsibility is to control movement of the bowels. Next, we have long reflexes. So no matter the origin, they're always going to be integrated in the central nervous system. They originate from sensory receptors in the GI tract or outside of the digestive system. And some of these reflexes are classic neural reflexes, where the stimulus is received and transmitted to the central nervous system along a sensory afferent neuron, while others originate outside of the GI tract. These are called cephalic reflexes, and it's called cephalic because it originates in the brain. And there are two types of cephalic reflexes. These include feed-forward and emotional reflexes. So feed-forward reflexes begin with a stimuli such as sight, smell, sound, or even thought of food, and it prepares the digestive system for the food that the brain is anticipating. So if you smell or see someone cooking bacon, your mouth will probably be watering and your stomach will start to growl. And emotional reflexes occur when the GI responds to a range of emotions. So this can include butterflies in the stomach when you're nervous, traveler's constipation, and even psychologically induced vomiting or diarrhea. And smooth muscle and glands of the GI tract are under autonomic control. So this includes sympathetic and parasympathetic neurons. So the sympathetic neurons inhibit the GI function, and parasympathetic neurons are excitatory and enhance GI functions. Most parasympathetic neurons are found in the vagus nerve. So here we have a figure that I took from the Silverthorne text, and it depicts what we have just gone over. So it shows the enteric nervous system right here, the little brain, and the sensory receptors and neurons, interneurons, and enteric neurons. And the red arrows are the short reflexes. So we discussed that the little brain is mainly consumed of the short reflexes. And then the long reflexes right here are shown up here with the cephalic phase or feed-forward stimuli going down sensory receptors in cephalic brain to then stimulate the enteric neurons of the little brain. Then we have over here the local stimuli, so the distension of the stomach, presence of food, osmolarity, and acid or pH levels. I think this is a really good figure to study when trying to get an overall depiction of what the enteric nervous system entails. So next I'm going to talk about the integrated function within the enteric nervous system. So the cephalic phase is the first phase that starts digestion. It has long reflexes that begin in the brain and create a feed-forward response. So like we discussed earlier, the anticipatory stimuli activate neurons in the medulla, and then the medulla sends efferent signals to through the autonomic neurons to salivary glands, and then the vagus nerve to the enteric nervous system. So salivation is the first phase of the cephalic phase, and it serves 4 functions. It is to soften and moisten food, digestion of starch, taste, and defense. And next we have mastication, which is just chewing and softening the food to make it easier to swallow. And then the deglutition, which is the act of swallowing, and it's a reflex action that pushes food or liquid into the esophagus. We have the gastric phase, which is a series of short reflexes in the lumen of the stomach, initiated by distension or peptides and amino acids. This activates endocrine cells and enteric neurons. The gastric secretions protect and digest. So right here I have another image from Silverthorne, and it shows the secretory cells of the gastric mucosa. So right here you can see the type of cells and then what substance is secreted and function and stimulus for release. So it just shows the different ways that the stomach secretes to protect and digest, perform its functions, basically. So next we have the intestinal phase, and so this begins once chyme passes into the small intestine. So chyme is a pulpy, acidic fluid consisting of gastric juices and partly digested food. So this comes from the stomach in the gastric phase, and then it comes into the intestine to begin the intestinal phase. And it begins and mainly occurs in the duodenum of the small intestine. So this is just the first part of the very beginning of the small intestine. So the stretching of the duodenum triggers vagal reflexes, and peptides and amino acids in the chyme stimulate the G cells of the duodenum to secrete more gastrin, further stimulating the stomach. So next, the acid and semi-digested fats trigger the enterogastric reflex. This is where the duodenum sends inhibitory signals to the stomach via the enteric nervous system. It also sends signals to the medulla that inhibits the vagal nuclei. So doing this decreases the vagal stimulation of the stomach and stimulates the sympathetic neurons that send inhibitory signals to the stomach. It's important to note that stretch activates short reflexes via my enteric nerve plexus, and the stretch activates the long reflexes via the vagus nerve and brainstem. And that is all I have. That will conclude my discussion of the short and long reflexes of the enteric nervous system. Thank you for listening.